Amyloid proteins are a protein that form fibril aggregations in the form of highly aligned beta sheet through the self-assembly process through interactions between proteins. These fibrils are toxic and are known to be associated with the development of amyloidogenic disease. A lot of efforts has been made to track the structural dynamics of amyloid proteins during complex aggregation process using spectroscopic techniques such as IR spectroscopy. However, in IR studies, use of water has been limited due to the spectral overlap problem of amide 1 band, which provides information on secondary structure of the protein and the bending mode of water. Therefore, most of the protein IR studies use heavy water. However, the use of heavy water raises the question of whether the observed protein dynamics when using heavy water provides information of the real biological environment. In this study, we overcome the problem of using heavy water by using two-dimensional infrared spectroscopy. 2D-IR is a spectroscopic method that measures the tertiary signal derived from 3 IR pulses with the time difference. Through 2D-IR, structure information and dynamics can be observed by the position and intensity of diagonal and off-diagonal peak and peak difference as waiting time changes. In particular, since 2D-IR has high spectral and temporal resolution, analyzing between modes with large difference in transition dipole strengths and vibrational lifetime becomes easier. And using this advantage of 2D-IR, only the protein signal can be observed without the water signal by setting the appropriate concentration and waiting time when measuring the 2D-IR spectrum. Therefore, in this study, we aim to understand the difference in amyloid aggregation dynamics in water and heavy water using 2D-IR. Insulin was used for the study because it shows evident secondary structural change during the fibrillation process. The insulin fibrillation process was accelerated through incubation, and samples were extracted at regular time intervals to observe changes during the fibrillation process. First of all, we confirmed the difference in the fibrillation mechanism and kinetics of insulin in water and heavy water through thioflavin T fluorescence and TEM image. These two results indicate that heavy water only delays the nucleation and elongation of insulin but does not inhibit fibrillation formation itself. The reason for this difference in water and heavy water could be explained through 2D-IR results. To obtain a protein 2D-IR signal in water, it is important to set appropriate waiting time. Through scanning the waiting time, 250 femtoseconds was set as appropriate waiting time because water signals appear at short waiting time and thermal response signals appear at long waiting time. As follows, we measured 2D-IR spectra of insulin during fibrillation at a waiting time of 250 femtoseconds. At early time, a peak corresponding to alpha helix, which is the native state structure of insulin monomer, was observed. As fibrillation occurs, a peak corresponding to beta sheet, which is the structure of formed fibril, was observed. Interestingly, due to improved spectral resolution of 2D-IR, two more peaks were observed in the early time of fibrillation in heavy water. These different spectral line shapes of the initial spectra of water and heavy water means that insulin has a different initial structure in the two solvents. These two additional peaks in heavy water can be assigned as a beta-strand peak that appears when insulin exists as dimer. This results means that insulin exists majorly as a monomer in water and a dimer in heavy water. With additional experiments using SOX, MD simulation, and DSC, we could explain this structural difference is due to the solvent isotope effect of heavy water. And we could conclude that increase of dimer formation of insulin in heavy water induces a retarded structure transition required for subsequent protein nucleation process at an early stage of fibrillation. These results confirm the importance of using water as a physiological solvent in dynamic studies related to protein misfolding and amyloidosis using various IR spectroscopy and microscopy.